Apparently my brain's not functioning, but that's okay. Let's go ahead and talk about water. One of my favorite substances. I'll tell you why, for several reasons. In my younger days, I used to work in restaurants, and in my household, when I was growing up as a young boy, we had to wash our own dishes. We had a dishwasher, but unfortunately it didn't work. So 3 of us children had to wash dishes. Long story short, I learned the value of soaking pots and pans and dishes. And that value is because of the properties of water. So we say that water is the universal solvent, geez. All right, and so what we mean by water as the universal solvent, okay, solvent is going to be a substance in which you can add another substance to, and it will dissolve that second substance that you added to whatever the initial substance is. So for example, water will be our solvent. If I add salt to it, you should see the salt dissolve into the water. So when we break it down, okay, when we add certain substances to water, there are substances that don't dissolve in water, and there are substances that do. So substances that are non-polar molecules will not dissolve in water. So we call those water-fearing or hydrophobic, like oil. And some of you have boiled pasta before, and I'll usually, when I make pasta, I'll add a little bit of oil to the water. And so you'll see that oil kind of coagulating on the surface of the water. And that's because, right, it's hydrophobic. It will not dissolve into the water. Now, substances that do dissolve in water, right, we call those hydrophilic. They're water loving. And those are going to be polar molecules or ions. Right, so we want to keep in mind these properties. And so when we add these substances to water, okay, they'll disperse and pretty much break down into where we can't see it anymore. So those are going to be polar molecules and ions, which brings us to our next topic, which is going to be talking about acids and bases. So, but let's not jump into that yet. So you'll notice the title of this slide is called Water, a Neutral Solvent. Okay, and so what that means is water, if you have a cup of water on your desk right now, right, water will spontaneously, for no apparent reason, it's just going to break apart. Now we know, sorry, I see someone asking a question. Can I go back for a moment? Absolutely, yes, I can. There you go. So we know the molecular formula for water is H2O. All right, so H2O has 2 hydrogen molecules and 1 oxygen. And so what will happen is that water will spontaneously break apart, or what we call dissociate, into 1 hydrogen ion, and we call it an ion because it's charged, and you can see that little plus sign there, and 1 what we call a hydroxide ion. So the hydrogen ion is marked there with an H+, right? And then our hydroxide ion is written with an OH with a negative side. But if you notice, when water disassociates, when it just breaks down spontaneously, we're going to wind up with 1 positive ion, or 1 cation, and 1 negative ion, or an anion. So if you have 1 positive charge and 1 negative charge, what's going to happen? What do you think is going to happen? You have 1 positive charge and 1 negative charge. They'll cancel each other out, right? So here's that formula that I just drew on the previous page. I want to walk you through this slide. Okay, so keep in mind, this is what's going to happen when water spontaneously disassociates. All right, when it dissociates, which is what's going on here, Right, that's how we're signifying that. All right, it's going to form ions, and it's going to form one positive ion and one negative ion. All right, so when it breaks apart, we're going to find out that we're going to wind up with equal numbers of positive hydrogen ions and equal numbers of negative hydroxide or hydroxyl ions. And you can see that written down here. All right, so what will happen is now this hydrogen ion is going to go and look for a water molecule, and it's going to add itself to that water molecule. And it's going to form what we call hydronium. And that's this guy, H3, but still it's got a positive charge, and we'll still have our hydroxide ion down here. So the point of this slide is to show you while water is spontaneously just breaking apart and dissociating into these ions, it's going to create equal hydrogen positively charged ions and equal negatively charged hydroxyl ions. All right, so those charges will cancel each other out. And that's why water is a neutral solvent. Okay, this is going to bring me to my next point here. So bear with me on this. So that brings us to acids and bases. So now, instead of talking about water, let's talk about an acid. So what is an acid? An acid is going to be something, a substance, that when we add it to water, right, it is going to produce, right, a hydrogen ion, a positively charged hydrogen ion, and a negatively charged anion. So there's another name for an H+. You can call it a hydrogen ion, but we also call it a proton. I know, go figure. We just talked about protons and neutrons and electrons, right? So now this is something else that you have to memorize. All right, so a proton is another name that we give the H+. So when I add an acid to water, that acid is going to dissociate. Okay, so for example, hydrochloric acid, HCl. When I add hydrochloric acid to water, it's going to break down into my hydrogen ion or my proton and an anion, which in this case will be, sorry, I don't know why it's doing that, chloride right here. Okay, so we have one positively charged ion, which is going to be the proton, and then we have a negatively charged ion or anion, which is the chloride when we're talking about stomach acid. Or I shouldn't even say stomach acid, excuse me, when we're talking about hydrochloric acid. Okay, so substance A, which is going to be our acid, when we add it to water, is going to dissociate into our hydrogen ion or the proton and an anion. That's the definition of an acid. Okay, so acids, and this is important, are going to, when you add them to water, they're going to increase the concentration of free hydrogen ions. Which makes sense, right? Because when we add the acid to the water, it's going to dissociate and break down into a hydrogen ion and an anion. So stronger acids will see a lot more dissociation. It's real easy for them to break apart. Weaker acids, it's not as easy. Okay, so we'll see less dissociation in weaker acids. So more dissociation in stronger acids, and basically that just means the acid just breaks down into the proton and the anion. And that's what we're going to see. Now, when we're talking about acids, we also have to talk about bases. And a base, I want you to think like a sponge. Right? Our bases are a sponge, and what they do is when we add a base to a solution, it's going to go around that solution and it's going to look for these hydrogen ions that are all by themselves. and it's going to go around and suck them all up or accept them. So we call bases proton acceptors. So when you add a base to a solution, it's going to go around that solution and soak up all those free floating hydrogen ions. So essentially, a base is going to decrease the concentration of those free hydrogen ions because it's going to go around and suck them up. It's going to absorb them. So stronger bases, we're going to see more absorption of the hydrogen ions. Bleach, ammonia, that's why they make such great cleaners, folks, because they're going to go around and suck all this stuff up. Our weaker bases, right, they are less likely to absorb. But there's a time and a place for that, and we'll talk about it when we get into, all right, some of the buffers in your blood. The buffers in your blood help to prevent major changes to the acidity of your blood. Okay, bicarbonate is an example. We'll get into more of that. So basically, when we're talking about a base, when you add substance B, which stands for base, to water, it's going to go around and suck up all the as much of those free floating hydrogen ions in the water. Okay, so think of it like that. So here's a nice question for you. What type of substances release hydrogen ions when they are added to water? Okay, if you can, type in your answer in the chat bar, but I'm not going to wait all night. Okay, so let me repeat the question. What type of substances will release the hydrogen ions when we add them to water? And I'll give you a hint. The answer is on the screen here. All right. I see lots of answers coming in. I love you guys. This is right. Yes, yes. Acids, acids, acids. Good, good, good. Acids. Correct. So our acids are going to be our proton donators. They're going to donate. They're going to release their hydrogen ions when they're added to water. Good, 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 good. Okay, so that brings me into now, we're going to start talking about acids and bases. So there's a couple things that we need to first look at. We need to first look at assigning a value to something that's acidic, neutral, or basic. So we've come up with this thing called the pH scale. And so when we talk about pH, basically what we're doing is we're measuring how much hydrogen ions are going to be found in this solution. All right, so the scale that we've come up has a range of 0 to 14. And right in the middle of this scale is water, plain water. It has a value of 7. So it's right in the middle. And remember what we said about water. I'm going to go back to this slide. I'll come right back to this. Check it out. Water is a neutral solvent. Okay? So water is neutral. And the reason why it's neutral is because it's always going to produce equal numbers of a positive hydrogen ion and equal numbers of negative hydroxyl ions. Okay, so keep that in mind. So that's why we put plain water right in the middle, because when it dissociates, it's always going to make equal numbers of these guys right here. So one positive charge will cancel out one negative charge. All right, so here's the next concept that I want you to understand. When we talk about the pH value and the concentration of the hydrogen ions in a solution, right, those two values are what we call inversely related. Real easy to remember. When I say something is inversely related, that means if the value of one of those two things increases, then the value of the other thing has to decrease. As one goes up, the other goes down, and vice versa. So here's what this means. as the concentration of hydrogen ions increases, the pH value will decrease for a solution, and vice versa. As the hydrogen as the hydrogen ion concentration decreases, our pH value for that solution will increase. All right, I'm going to walk you through this. Okay, don't worry if you're not like understanding where Dr. Kaz is going with this right now. Okay, so a couple things. pH value. Okay, when we look at the pH of something, we're seeing, all right, it is the amount of hydrogen ion in that solution. Okay, that's one thing. The other is plain water is right in the middle and it has a value of 7. So in the middle of our scale of 0 to 14, if you have a value of 7 for something, you're neutral. And then the last thing is that the pH value and the concentration of hydrogen ions in a solution are inversely related. So if I increase the amount of hydrogen ions in my solution, the value on my pH scale of that solution goes down. And I'll tell you what that means here in a moment. Right? And vice versa. As the hydrogen ion concentration decreases in my solution, the pH value of that solution increases. So how do I interpret this pH scale? No problem. Okay, so like I said before, solutions that when you add something to it, okay, if it gets equal amounts of hydrogen ions or hydroxide ions, they'll always be neutral because remember, they'll cancel each other out. So we assign them a value of 7. Now, if I take some solution, something, and I add it to water, okay, and I find out that I get way more hydrogen ions than I do the hydroxide ions, then that solution is going to be acidic. Because remember, acids donate hydrogen ions. It's a proton it's a proton donator. H plus is a proton. So when I add my substance to the water, right, and I find that it's just blowing out tons of hydrogen ions, way more than the hydroxide ions, then I say, okay, that solution is now acidic because of all of these hydrogen ions, and it has a pH value of less than 7. So anything that has a pH value of less than 7 is considered to be acidic. Okay, now, if we flip it, and if I now take something and add it to the water, and I find out that I get way more hydroxide ions and very few hydrogen ions, then that solution is basic or alkaline. And it has a pH value of greater than 7. So anything with a high pH of greater than 7 is basic. Anything that has a low pH that's going to be below the value of 7 is going to be acidic. And when we go from one value to the next, so if I go from a pH value, all right, from 7 to 6, all right, I'm now less than 7. Okay, so that means that that solution is acidic. There's a 10 times greater concentration, right, of hydrogen ions in my pH value of 6 compared to 7. Point being is, as we move from one number to the next, it's a 10-fold change. That's what you need to know. As you go from one increment to the next, that's a 10-fold change. All right, so to recap, if you have a pH value of 7, you're neutral. If you have a pH value less than 7, you're acidic. And that means my solution will have a lot more hydrogen ions, positively charged hydrogen ions, than my negatively charged hydroxide ions. If I have a pH value of greater than 7, then my solution is going to be basic or alkaline, same thing. And that tells me that I have far fewer hydrogen ions floating around in my solution than the hydroxide ions. So that's why I love this picture here, because this kind of shows you what we're looking at. All right, so if you glance at these 3 beakers here in front of us, we have an acidic beaker, we have a basic beaker, and we have a neutral beaker. So if you were to look at the neutral beaker first, and if we were to go through and count the hydrogen ions and the hydroxide ions, they would all be equal. You'd have the same number of H pluses as you would with OH negatives. That's that neutral value, and so that is 7, which is pure water. All right, so if we look here now at this top beaker, if you go through and count up all the OH negatives, the hydroxides, 1, 2, 3, and 4. And then if we look at our positively charged hydrogen ions, our protons, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, looks like 11 or 12. All right, there's far more hydrogen ions than our hydroxide. So when you see far more hydrogen ions than the hydroxide ions, we are going to be acidic. And so we are going to have a low pH value. Check it out. See our pH values of 6, 5, 4, 3, 2, and 1. These are all going to be acidic because we have way more hydrogen ions in our solution. So you can see some of the values here. Hydrochloric acid. Your stomach is going to be secreting that, right, into the digestive enzymes there to break down food and certain liquids. All these here are going to have acidic properties. Now, if we go to our beaker here at the bottom of the screen, if we go through and count up all the positively charged versus the negatively charged hydroxide ions, we're going to have far fewer hydrogen ions compared to a numerous amount of hydroxide ions. So you'll see when you have far less hydrogen ions compared to the hydroxide ions, right? Essentially saying we have very little or few hydrogen ions in our concentration here, we're going to assign them to a higher pH value. And so they're going to be basic. Okay. Seawater, antacids, perfect. Antacids, what do we use those for? To help deal with neutralizing acids when you get an upset stomach. We'll talk about that. Okay, so keep that concept in mind when we're talking about the inverse relationship between hydrogen ion concentration and between the pH scale. As one goes up, the other it goes down. So that brings me to the important concept of neutralization and buffers. So when we look at neutralization, all right, we either have a solution that's acidic or basic. And when we neutralize that solution, what we're going to do is we're going to return whatever its pH value is to neutral, which means we need to return that pH to 7. So if it's acidic, it can have a pH value of 5. If it's basic, it could have a pH value of 12. So what do we need to do, all right, to get it to the neutral value of 7? Simple. If our solution is acidic, we just add a base to it. And if our solution is basic, we just add an acid to it. That's how we can neutralize stomach acid. Okay, stomach acid has a low pH. So in order to increase the pH value from 2 to 3 up to 7, we add a base to it. That's what an antacid is. It's going to go and interact with whatever that stomach secretion solution is, and it is going to start going around and absorbing any of those hydrogen ions, returning our pH value hopefully to 7. Neutralization, okay, simple. If you're too acidic, we're going to add a base to you to make you neutral. If you're too basic, we're going to add an acid to you to make you neutral. Now let's get to buffers, because buffers are hugely important. You'll learn more about their the importance of them when you take Bio 211. But what buffers do is they help to prevent pH changes to begin with. See, with neutralization, the pH has already changed. It's no longer 7, for example. It is either basic or acidic. And so now we got to get it back to neutral. Well, with buffers, we're trying to prevent the pH value from changing at all. Okay? So we want to always kind of keep with buffers, all right, our pH within an acceptable range here. And I'll show you what that range here is in a moment. So what will happen with buffers is something will happen in which, for example, all right, the pH of your blood in the human body is, the range is 7.35 to 7.45. So actually what we're looking for is a pH value of your blood of 7.4. That's the set point. That's the middle number. And they can float around in that range. So your body's always, you can, if you notice, that's slightly basic. Okay. So we want to float our blood pH around, all right, that value there at all times. Well, of course, as you know, something's going to happen, and we can either add excess acid or an excess base to your blood. And that will start to change the pH of your blood. Well, we have to keep it at this value, because then bad things will happen. You'll learn about some of these bad things as the course goes on. So what we're going to try to do is with buffers, these buffers are going to run around and they'll go ahead and accept, excessive hydrogen ions to prevent, all right, your blood from becoming too acidic. Or vice versa, these buffers will go ahead and donate hydrogen ions, right? Because they're always going to try to neutralize whatever it is that has been added to your body to change your pH. So in your body, you have these buffers, carbonic acid and bicarbonate, that float around in your blood that help to maintain that 7.4 pH value. And what they'll do if it starts to swing and become too acidic, okay, these buffers will then start to soak up, all right, those hydrogen ions. If it starts to become too basic, no problem. All right, these buffers will start to donate these hydrogen ions, all right, to swing, all right, the pH value of your blood into that range there of 7.35 to 7.45. And this is constantly happening all the time. So what is the general relationship of hydrogen ions, protons, H+, and pH? And this is that inverse relationship where I was telling you about. So with a greater hydrogen ion concentration, the pH goes down, and vice versa. With fewer hydrogen ion concentration, the pH value goes up. If you don't believe me, look at this picture here, and you can see our pH values on our scale. And then you can look over here in these beakers and see how many hydrogen ions are in that top beaker, which is acidic. A lot. So the pH value is low. If you look over here at this beaker here, you'll notice there's very few hydrogen ions in that solution. And so the pH value is quite high. Okay? Don't worry if you're not grasping all this right now. You will get this. I'm telling you, it will happen. You just got to go over this material. All right. I know that acids and bases aren't terribly exciting for some folks to talk about, but macromolecules are. All right, especially for folks that are interested in going into nutrition, because you hear this quite often nowadays when people talk about counting their calories and having to take in certain types of carbohydrates, proteins, fats into their diets. That's what we're really getting into, are these biological macromolecules. Macro means large. These are just larger molecules. So let's talk about these molecules here, because now we're starting to move up our organizational ladder here. Okay, we started with atoms, All right, and molecules. Now we're going to start to get to these bigger molecules, because these bigger molecules are going to start to form structures, organic structures. So speaking of organic structures, let's talk about organic versus inorganic molecules. You heard me mention this before. Right? Organic molecules are any molecule that contain carbon. Plastic is carbon, right? Plastic has carbon in it, so it's organic. We're obviously organic because several of the different molecules in our body contain carbon, okay? So that's what we're going to see. You're going to see in living organisms, all right, carbon in the molecular formula here. So when we get into these macromolecules or these biomolecules, we're going to learn about all these different things here. Now, inorganic molecules, real easy, are all the other molecules. So if you don't have carbon in your molecular formula, you're going to fall in the inorganic molecule category. So when we talk about the 4 classes of these organic molecules or these biomolecules, we're going to talk about lipids, which are fat, carbohydrates, okay, nucleic acids, and proteins. So now we're going to start to kind of shift into that fun stuff here where we get to talk about, all right, some of these macromolecules that you might be interested in. All right, so
can you go back for just a second?
Pardon?
Can you go back for just half a second?
Sure. Yep. So that's what we're going to be talking about now. We're going to go over some of these various molecules here and discuss. All right.
Thank you.
Yep, which is largely important. So I got to go down and just go through some of the basics here, and then we'll get into what I call the good stuff here. So these biological macromolecules are made in our bodies, and they have to contain carbon, obviously, right, because they're organic, hydrogen, and oxygen. So C, H, and O. They have to contain those. And then some of our macromolecules will contain nitrogen and phosphorus or sulfur. Okay, but all of them have to contain carbon, hydrogen, and oxygen. So we're going to start off by talking about what we call the carbon skeletons. And these are kind of like the backbone structures of some of these molecules. So you'll hear the term hydrocarbons. And if you hear that term, then without even looking at the molecular formula for whatever, you will know that the only atoms that structure contains are carbon and hydrogen. Hydrocarbons only contain carbon and hydrogen atoms. Okay, just in case you hear that, you need to know that. So when we're looking at these macromolecules, parts of these molecules are going to contain what we call functional groups. And these functional groups give a lot of properties to these items. All right, but a couple characteristics that when we're talking about these functional groups is, one, they are going to be polar. All right, and these functional groups are going to form, or have the capability, I should say, of forming hydrogen bonds, which again is important when we get into organic chemistry, biochemistry phenomena. Again, this is not an organic chemistry class, and I'm not going to go into that. But when we form these hydrogen bonds, it's going to allow for certain chemical processes to occur in our bodies. So some of these functional groups, right, can act like acids. Some of these other functional groups can act like bases. So if you, and again, I'm not expecting you to be able to identify what these groups are when you're looking at a structural formula or a molecular formula, but just something that you should know, that a carboxyl group, if you see it, right, on a molecule, that will act like an acid. Whereas if you saw an amine group, and I'll show you some of these, that will act like a base. Again, I'm just giving you some real basic information. All right, so just the important thing is these functional groups are going to really provide us with the reason why these molecules function or work the way they do. So let's talk about lipids, fats. Okay. First thing, when you think of lipids, think of a fat, but you need to also consider that lipids are water insoluble. What does that mean? That means that those are hydrophobic. They will not dissolve in water. Right, so keep that in mind. That's an important property to understand when we're talking about lipids. You know, oil and water just don't mix. Right, so they are water insoluble molecules. That's important. We come back to when we circle back through all these structures here. Okay, so the functions of lipids you need to know. Right, we'll use lipids for energy. Okay, we store lipids like triglycerides in our fat cells. And when we're in a situation in which we need energy, right, our body will start to break down those lipids in those fat cells, and then we can convert it into ATP, energy. Also, we're going to see lipids in the plasma membrane of our cells. That's the outer shell, I shouldn't say shell, but the outer envelope of a cell. If you don't even know what that is, no problems. When we get to chapter 4, we're going to talk about all that fun stuff. All right. We'll also use lipids to help make hormones. What are those? Chemical messengers. Lipids are also used for, or fats also used for cushioning, insulation. So when we talk about lipids, there's 4 classes. Triglycerides, which is the most abundant, all right, energy source. Your body would love to use triglycerides because you get the most bang for your buck. Then we have phospholipids. Then we have steroids. And then finally our acostanoids. So those are the 4 classes of our lipids: triglycerides, phospholipids, steroids, and acostanoids. We're going to go through each of these right now and talk just a little bit. So let's look at the first one, triglycerides. Okay, again, triglycerides, energy. All right, so your body is going to take triglycerides, all right, and use them for energy. So what is a triglyceride? Easy. A triglyceride is 1 glycerol group and 3 fatty acids. Now, don't worry. Let me see if I got the slide in here. Yes, I do. All right, I will show you what all that means. But for right now, just write this down. A triglyceride is a glycerol group. We also refer to the glycerol group as the glycerol backbone. And then it will have attached to it 3 fatty acids. They're these long chain-like structures that come off like that. And those fatty acids can vary. They don't have to all be the same length. Okay, some might be longer than others. So when we talk about fatty acids, right, a couple things, properties you want to know. They will vary in length and they will vary in number of double bonds. Dr. Kaz, what the heck is a double bond? Remember from last class, covalent bonds? Okay, so when we're talking about double bonds, we're saying that there are 2 pairs of electrons being shared between these molecules. And so for right now, just think, all right, the more double bonds you have, the more energy you can get. Triple bonds have lots and lots of energy. All right, so when we're describing something as a saturated fatty acid, for example, that means there are no double bonds. Okay, when you hear that term, saturated fatty acid, then you should immediately think, okay, there's no double bonds in this molecule. If we say it's an unsaturated, all right, fatty acid, then it has 1 double bond. Poly, unsaturated means many. So that means it'll be 2 or more double bonds. That's it. Okay, those 3 terms, that's simple. If it's saturated, we've saturated all the bonds, which means there are no double bonds. If it's unsaturated, there's 1 double bond. If it's polyunsaturated, then that means that there's 2 or more double bonds. All right, those are our triglycerides. So here, we're talking about our triglycerides, all right, you need to know it's the most common form of lipid. We will use it for energy storage. Where do we store it? In fat tissue, or what we call adipose tissue. All right, we'll also see it, some of the functions for structural support, cushioning, and insulation. And then what is a triglyceride? Look at the word, tri, 3, okay? Has 1 glycerol molecule and 3 fatty acids attached to it. So the job of our fat tissue, our adipose tissue, is to store these triglycerides for a rainy day when we need it, okay? So when you have excess nutrients in your blood, you just ate a meal, a nice big whatever meal, your favorite meal, I don't care, okay? But you eat plenty of vegetables, you're eating plenty of carbohydrates and a protein of some sort, all right? Either tofu, meat of some sort, something that's going to be high in protein, okay? So when you start to digest this, all of this stuff that you put into your mouth that winds up into your stomach, hopefully your body's going to break it down and then put it into the blood. And then it's going to circulate it throughout the body. And the tissues that need whatever these nutrients are will just take it. So when we have lots of nutrients floating around, okay, your body wants to save that for a rainy day. Once it satisfies all the immediate energy needs, then it says, oh, I got all this extra. What do you do with your leftovers? You put them in the refrigerator and you'll heat them up later on. That's what's going to happen here, okay, with triglycerides. All right, so you have all these nutrients floating around in your blood, and then your body's like, look, I don't want to get rid of this stuff. I want to save it for later. So I'm going to make these triglycerides and then store them in my adipose tissue. When it does that, all right, it undergoes the process of what we call lipogenesis. Genesis means to create, to make. Lipo stands for lipids, fat. So if you dissect this word, this is making of fats, and triglycerides are a lipid. So again, we make these triglycerides from all this excess nutrients that are floating around your blood because you just had this awesome energy-rich meal. Now, say you haven't eaten in days, and your body's like, look, you're not putting food in your mouth and in the stomach. I got to get, some energy from somewhere. So it is going to then go back to that adipose tissue where those triglycerides are stored, and it is going to release those triglycerides from the fat tissue and put it back into circulation. So that process, when we break down those triglycerides into our glycerol molecule and fatty acids, that process is called lipolysis or lipolysis. Point being is, dissect the word. Lysis means to break down. And then we already know what lipo is. We're going to break down the fats. And that's what happens. So when you got lots of energy floating around or nutrients floating around in the blood, we're going to store it for a rainy day and undergo lipogenesis. When you're in need of nutrients, okay, we got to free up some of that stored energy. All right, no problem. Our fat cells are going to go and then start to break down the triglycerides and release it into the bloodstream to increase those nutrients so the cells and the tissues that need, all right, that those molecules or whatnot, or those substances have it readily available. And that's what this picture is showing you here. Here's your triglyceride molecule here at the top, right? You've got your glycerol backbone right here. And then you got your 3 fatty acids coming off. Okay, and so basically, depending on what you're undergoing, okay, whether it is lipogenesis or lipolysis or lipolysis, is going to determine what you're going to do to the triglyceride. If energy levels are low, or excuse me, if nutrient levels are low, then you're going to undergo lipolysis, and we're going to liberate those triglycerides. If you got lots of nutrients floating in your blood, and all that. Then we're going to store that stuff for a rainy day, and we're going to undergo, all right, lipogenesis. All right, our second type of lipid is going to be phospholipids. Phospholipids, all right, are in every single one of your cells because they make up a good portion of your cell membranes. We'll talk more about that in chapter 4. So phospholipids are what we call amphipathic molecules, which means these molecules have a polar component and a non-polar component. Okay, so phospholipids are an important part of our cell membranes. We need these, okay? And so they're amphipathic, polar and non-polar components here. So they're going to look similar to a triglyceride, except, right, we're only going to have 2 fatty acids attached to it. All right, so one end of our molecule is going to have that glycerol group there. And then on the other part, we're going to have 2 fatty acids. So what makes our phospholipid polar and non-polar? Right? The glycerol phosphate and any organic groups are going to give phospholipids their polar attribute. The fatty acids will give it the non-polar. And that makes sense because if something's polar, it can dissolve in water. All right, but if it's non-polar, it will not dissolve in water. So if you're polar, you're hydrophilic, water-loving. We can dissolve you into water. But if you are non-polar, you are hydrophobic, water-fearing. You will not dissolve in water. All right, that's the phospholipids. We'll get more detail, trust me. As this class or course goes on, we will get into more of these concepts in more detail. We're just going to kind of give you a nice kind of brief exposure, surface exposure here. All right, so the third type of lipid are steroids. And a lot of us have heard steroids, but we have no idea what a steroid is, okay? A steroid is a multi-ringed structure. You will actually see 4 carbon rings. And out of those 4 carbon rings, 3 will have 6 carbons, and then 1 will have 5 carbons. And this is important because this helps us to set up this molecule here, cholesterol. We're all familiar with cholesterol. Cholesterol is actually, believe it or not, a much needed, all right, lipid. You'll find it in the plasma membranes of our cells. And I'll get into the benefit of having cholesterol in your plasma membrane. It actually will make the plasma membranes to these cells more tolerant of changes in temperature. All right, so it's important. But what we'll also see is that cholesterol is going to be found in our steroid hormones. So it's important. You need to have, all right, cholesterol, because it's an important ingredient for things like steroids, bile. In case you're not sure what bile is this substance that the liver makes. Your gallbladder stores it and concentrates it, but it helps you to break down fats. All right, so cholesterol is an important component for that. All right, so that's going to be steroids. We'll cover more of that in chapter 17. Lipids, the last type of lipid are the acostanoids. And sometimes we refer to acostanoids as locally acting hormones or local signaling molecules. So basically, acostanoids come from a plasma membrane component called arachidonic acid. Arachidonic acid is one of the components there in your phospholipids. And so what will happen is if there's damage to the cell, all right, arachidonic acid will undergo some chemical reactions. And as a result of these chemical reactions, it will produce acostanoids. What are the acostanoids? They're listed right down here. Okay, prostaglandins, prostacyclines, thromboxanes, and leukotrienes. They have many, many different properties here, right, which I'm not really going to get into. But what you do need to know is they do play a role in inflammation. So these acostanoids are going to be found at different parts of the inflammatory process. And so what will happen is certain drugs can block the formation of these acostanoids and help to decrease inflammation and can help decrease pain. But again, we're not here to really get into that. But what I want you to know is these acostanoids come from arachidonic acid. Where is arachidonic acid found in? It's found in your plasma membranes of your cells, every single one of your cells. Okay, so arachidonic acid comes from the phospholipids there. And so what will happen is that arachidonic acid will undergo these chemical reactions and it will form the inflammatory response or whatever. And it can turn into any of these four classes: prostacyclines, prostaglandins, thromboxanes, and leukotrienes. Okay. So when we're looking at fats, just to kind of go and throw a little bit of a clinical view at you, you've probably heard of saturated, unsaturated, and trans fats. Trans fats have been pretty much outlawed in areas of New York State. Literally in New York City, I believe they've outlawed all the trans fats. This is many years ago. But when we're looking at our fats, for example, animal fats, right? Animal fats are going to be saturated fats. What does that mean? There's no double bonds. So an animal fat, pretty much when it's at room temperature, will appear solid. Okay, like butter is solid at room temperature, at least it's supposed to be. Now, when we talk about vegetable fats, that's when we get into unsaturated fats. So these fats, when we're at room temperature, will be liquid. So basically, when you talk about fat, for the most part, a fat is going to be something that's going to be solid, and an oil, which is still a fat, is going to be something that's liquid. Really, to make it generally simpler for you, okay, fat is solid at room temperature, oil is liquid at room temperature. Obviously, all right, the unsaturated fats are going to be healthier, right? Generally, vegetable oil, olive oil, even coconut oil are all much better for you than butter, although butter tastes really good, okay? So what we can do is we can convert, all right, these fats, all right, into saturated fats through this process called hydrogenation. So we can make these oils solid, all right, through this process of hydrogenation. And because we do that, we can confer some of their healthier qualities. Now, there's a process called partial hydrogenation, and that's what led us to these guys, trans fats. Well, research and obviously much data collection has shown that trans fats are bad for us. So we don't use them anymore because they increase the risk of cardiovascular disease and stroke. So we don't mess with trans fats anymore. I recommend that if you have any, get rid of them. You don't want them. A couple other lipids, we have our glycolipids, and glyco just means we're going to attach a carbohydrate group to our lipid. And so this is important because, again, when we get into talking about the plasma membrane, which is that envelope that surrounds our cells, all right, these glycolipids are going to be important when we need things to bind onto our cell. All right, so glycolipids are basically a lipid molecule with a carbohydrate group attached to it, found in our plasma membranes of our cells, and it helps our cells to bind onto things. And then we have our fat-soluble vitamins, vitamin A, E, and K. fat soluble vitamin basically means that if you imbibe quite a bit of these vitamins, they can start to build up in your body's tissues, and you can actually reach toxic levels of these vitamins. Water soluble, you end up just peeing those out. So again, don't worry about when you look at this picture here, these major classes. I just kind of wanted to show you, all right, the different pictures of some of these different types of lipids. Okay, so here you can see the triglyceride with the glycerol group, and then the fatty acids coming off. Then our phospholipids, here you can see, here's part of our glycerol group with its 2 fatty acid chains coming off. Here's our steroid group here, all right? And then we have our acostanoids, our locally acting molecules here. Okay, so which class of lipid forms the cell membranes? I'm going to kind of move through this just a little bit, not because I'm behind, but I just don't want to rush you guys. All right, those are the phospholipids. Phospholipids are going to be found in your cell membranes. In the cell membranes. All right, let's move on to our next group of macromolecules, carbohydrates. Carbohydrates. All right, so what is a carbohydrate? All right, from a molecular standpoint, it is going to be a molecule that is made up of carbon, hydrogen, and oxygen, all right, atoms. So if we take this equation here, Ch2O inside of parentheses, and then outside the parentheses we have N. N stands for how many carbon atoms are present. So if you have 6 carbon atoms, then our molecular formula is going to be C6H12O6. That's our example. So in this case, when we're looking at carbohydrates, every carbon atom is either going to have, I shouldn't say either, is going to have a hydrogen ion, a hydrogen atom attached to the carbon, and a hydroxide or OH group will be attached to the carbon. All right, but let's get into the important stuff here. Not that that's not important, but stuff that's really going to affect you that you should know. All right, when we get into the carbohydrates, a lot of people think carbohydrates are sugars. All right, so let's talk about the different types of carbohydrates. We have what we call monosaccharides, disaccharides, and polysaccharides. Now, from the spelling of these words, mono means one, di means two, and poly means many. So if we're talking about a monosaccharide, right, that is going to be what we call our simple sugars. Glucose is primarily your body's favorite monosaccharide. Now, if we take 2 monosaccharides and we attach them to each other, we went from 1 sugar to now 2. So we call that a disaccharide. So all we're going to do is we're going to take 2 monosaccharides and connect them to one another. And then if we just start stringing monosaccharides to each other, then we have what's called a polysaccharide. I'll show you some examples here. Okay, monosaccharides, disaccharides, and then polysaccharides. So here is our most famous, most common monosaccharide, and that's glucose. Has 6 carbons. All right, so you know from the equation then what that means. When we're looking here, okay, at our example, that is the molecular formula for glucose. It has 6 carbons in it. So our chemical formula is going to be C6H12O6. So your cells of your body love to use glucose for energy. And so obviously, your body is going to make sure that the concentration of glucose in its tissues is going to be carefully regulated and carefully maintained. So now, what are we going to do if we have extra glucose? Do we want to keep it or get rid of it? Do we want to save it for a rainy day or just waste it? No, we want to save it for a rainy day. All right, so you just had a big meal, your blood sugar levels shoot upwards. So your body, all right, says, all right, we've used the glucose, all the tissues, all the cells are happy. They got all the energy they needed, but I got this extra glucose lying around. I want to save it for later on. Well, no problem. We're going to store glucose in tissues, in certain organs of our body, and we're going to store it in a substance that we call glycogen. So your liver and your skeletal muscles are going to take in the extra glucose, and it's going to store that glucose, all right, as this molecule that we call glycogen. So when you see glycogen, just think that that's the stored form of glucose. And so the process of making glycogen from glucose, we call that glycogenesis. Genesis is to make. Glyco, all right, stands for glycogen. So we're making glycogen. How do we do that? We just string one glucose molecule, and then we add it to another, and then we add another one to that, and make a big long chain, right, of glucose molecules. We call that one glucose molecule is called a monomer. And so what we're going to do is we're just going to string all of these monomers together to make glycogen. So when we need that glucose, we're going to undergo the process of glycogenolysis. Lysis is to break down. So we're going to break down that glycogen. And so the liver is going to go over there to glycogen, and it's going to cut off, right, one of the glucose monomers, and it's going to release it into the blood. It might release one molecule, it might release 100, 1,000, whatever, okay? So the liver is going to go to the glycogen and start breaking it down. If, let's say, all right, we still need more energy, your liver can also undergo the process called gluconeogenesis. Genesis is to make, to create, all right? Gluco stands for glucose and neo stands for new. So the liver, in order to make glucose, it's going to go and look for what we call non-carbohydrate sources. That means non-sugary sources. It might want to use fatty acids. It might want to use amino acids. So it's going to use something that we can't find in our carbohydrates. So it's going to undergo this process called gluconeogenesis that only happens in the liver, right? And so it's basically just going to try to make more glucose from non-carbohydrate sources. So here you can see a picture of glucose and glycogen. Genesis means to make, to create, to build. So here you can see this glucose molecule on our left side of our screen, and then our right side of our screen, we can see glycogen. So this is a monomer. It's one glucose molecule. And so now, when we want to store that glucose for a rainy day, we're going to undergo the process of glycogenesis. And your skeletal muscles and your liver do this. And basically, they're just taking glucose molecules and just making a big long chain. Now, when energy sources are, when you need energy, all right, and you don't have food to get that energy from immediately, what your body will do is the liver will undergo what we call glycogenolysis. And it will start to cleave and cut off these individual glucose monomers and release them into the blood. and they'll circulate around, and whatever tissues that need the glucose can use it. The brain loves glucose. It's its fave. All right, so that's how we make glucose into the stored form of glycogen. All right, so here are some other types of the carbohydrates here, right? So glucose has isomers. You remember isomers, right? Those are going to be these molecules that have the same, all right, molecular formula, but their structural formulas are different, which means they have different chemical properties. So for our monosaccharides, our simple sugars, right, we have three types. Okay, we've got glucose, we've got galactose, and fructose. Those are our monosaccharides. Okay, and so those are the hexose monosaccharides, meaning they have 6 carbons. We have our 5-carbon monosaccharides. You should know this, called pentose sugars. We'll talk about this when we get into DNA and all that. We have ribose and deoxyribose. And then if we take 2 monosaccharides and stick them together, remember what I said, we'll get our disaccharides. So we take 2 simple sugars, we stick them together, and now we're going to form a disaccharide. And so there's 3 types, all right? We have sucrose, table sugar, lactose, milk, and then maltose. Those are our 3 disaccharides. Each disaccharide is going to have at least 1 glucose molecule. So every single disaccharide has 1 glucose molecule. All right. One type of disaccharide can have 2 glucose molecules. I'll show you here in a second. Here are our monosaccharides. If you look here, right, you've got galactose, fructose, and then it's not pictured here, but don't forget about glucose. So these are the isomers of glucose. And then we have our 5 carbon sugars, our pentose sugars, ribose and deoxyribose. So we are going to now make our disaccharides. So you'll notice each of these green molecules, that is a glucose molecule. So if I take glucose and add fructose to it, we get table sugar. If I take glucose and I add galactose to it, I get milk sugar. And then if I take 2 glucose molecules and add them to one another, you get maltose. Those are our disaccharides. Yes, 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 yes. All this will be posted. You don't have to worry. Yep, I will post this. I will send you guys the links too. All right. And then another type of carbohydrate, remember I said we have monosaccharides, disaccharides, and then polysaccharides. That's going to be, all right, the sugars that we have that are going to be 3 or more. And I just went over one of them in detail, glycogen. Okay. In animals, glycogen is the most common type of polysaccharide. In plants, all right, starch and cellulose. Now, starch we can break down into what we call complex carbohydrates and then down into the more basic, simpler types of carbohydrates. Cellulose, unfortunately, our bodies can't break it down too well because plants have a cell wall and we have a difficult time digesting that. So cellulose we call fiber, okay? And so that's the roughage that you might take in. And it's not digestible and helps to move things through. All right, so keep that in mind. Cellulose, it's a good source of fiber, which is good from a nutritional standpoint, okay? But we have difficulty breaking it down, right? So it's going to be a non-digestible component. Whereas starch, we can break that down. And then another type of carbohydrate are what we call our GAGs, or our glycosaminoglycans. glycosaminoglycans. And basically, again, these are just a big carbohydrate molecule, and we have an amine group. And an amine group is going to contain a nitrogen molecule, all right, and then hydrogen attached to it, NH3. Right. And so these are going to be important when we get into tissues, and I'll talk about some of the importance of the glycosaminoglycans. But they're just a large carbohydrate molecule that will have, all right, an amine group, which is a functional group. All right. We'll also see some of our glycosaminoglycans or our GAGs, all right, they'll have proteins attached to it. So we call those proteoglycans. So these are important, and we'll talk about them when we get into, all right, tissues in chapter 5. But they make up an important component to what we call our ground substance. And these molecules attract water. Remember what I said, water's a follower. It's going to follow things. So what is the repeating monomer of glycogen? And where is glycogen stored in the body? So the repeating monomer of glycogen, we should know this, is glucose. Remember? We just keep making a big long molecule by stacking one glucose molecule onto the next, onto the next, and on the next. And where is glycogen stored in the body? There are 2 places. You need to know this. You need to know every single thing on this slide. Right? It's stored in the liver and in skeletal muscles. Well, that makes sense for it to be stored in skeletal muscles, because that means that those tissues, your skeletal muscles, don't have to wait for glucose to be released from the liver and then travel through the blood and get to the skeletal muscles. The glycogen is already there. It's on hand. So those skeletal muscles can start to break it down on their own. They don't have to wait on the liver. All right, one of my favorite substances here. You know what, guys? No. We're going to stop here.